Hello everybody. This video will be about immunology, innate immunity, and with this video we open new and the last chapter in this term, immunology. Okay, immunology it's a study, it's a science. It's a science which study our defense protection against everything foreign and this defense will be against viruses, bacteria, protozoa, or even larger parasites like nematodes. And in addition, this protection will be against our or proteins in autoimmunity and against our own aberrant cells in tumor immunity. So what is immunity? In short, immunity it's a self-defense against everything foreign. With this defense, our immune system can recognize self elements from non-self environment agents and make a defense against everything non-self from environment or wrong self cell like tumor. Immune system, it's a complex of organs, immune system cells, molecular components, and barriers tissue that provide immunity. Immunity is protect against infection and invasion. Everything foreign from environment. Okay, immunity consists of three parts. So barriers, physical, chemical, and biological. It is the first line of defense. Acid on the skin and stomach, mucous membranes, secretions, nasobronchial cilia, and microbiota. The secondary line of defense is non-specific response innate immune cells plus cytokines, complement proteins, lysozyme, basic polypeptides, and a non-specific response is relatively rapid, but not specific. And thus not always effective. And the third line of specific response, delayed response, it's a response with adaptive immunity cell, cytokines, antibodies, and adaptive immune cells. Specific response is slower in its development during an initial infection, which are pathogen, but is highly specific and effective at attacking a wide variety of pathogens. So it is the third line of defense, and it is slowly in its development, but it's more efficient than non-specific response. Okay, so as I said, immune system consists of some organs and cells. First of all, let's talk about organs. So, okay, organs are divided into two groups, central and peripheral. Central, it's a bone marrow and thymus. And peripheral, it's a or secondary, also we can say, encapsulated formation, lymph nodes, Peyer's plaque, and spleen. And unencapsulated formation like tonsils, cryptopatches, appendix, and other lymphoid tissue. Okay, and what about cell of immune systems? They can be granular or agranular. Granular, it's neutrophils, eosinophils, basophils, mast cell, natural killers. And granular cell, it's a cell of, and also a granular cell, it's a monocytes, plus their tissue, progeny, macrophage, and dendritic cell. Okay, granular and monocytes, they are cell of innate immune response, non-specific. They recognize conservative molecular structure like PMP with PRR and also DMPC and stress molecules. Have high speed of immune response. And also cell of immune system are lymphocytes. They can be T or B. And T and B lymphocytes, it's a cell of the adaptive immune response. And they recognize antigen. They need to antigen sensibilization before they became active. And they have its specific their specific receptors. T cell have TCR T cell receptor, and B cell have B cell receptor BCR. And this receptors recognize various individual antigenic molecules, antigens. Adaptive immunity with T and B cells generated within a few days. And after the primary immune response, T and B memory cells are formed, which activate secondary immune response. Okay, so as you understand, in our body, we have two types of immunity, innate or non-specific and adaptive specific immunity. Innate, it's a first immunity, first immune response against everything foreign. And this kind of immunity is antigen independent, and there is immediate maximal response. No antigen specific. Exposure results is no immunologic memory. And specific immunity, we can also call it adaptive immunity. It's always antigen dependent. So this immunity is developed after antigen stimulation. There is a large time between exposure and maximal response, so it needs some time to be developed. And always specific immunity is antigen specific. So uh, it has a specific antigen stimulation, and it reacts with specific antigen. Exposure results in immunologic memory. Immunologic memory, it's a result of specific immunity. Um, Innate immunity is an antigen non-specific defense against a mechanism. It's an antigen non-specific defense mechanism that a host use immediately or within several hours after exposure to almost any microbe. This is the immunity of one is born with, and is the initial response by the body to eliminate microbes and prevent infection. Okay, so in some literature you can find the physical defense. It's a physical and chemical defense. It's one part of immunity. It's like a first defense, and the secondary defense is the cellular defense. But sometimes, and more often, we can talk that it's all this, all this parts of innate immunity. So physical defense, it's like a barrier, like a skin, mucous tissue, and they said that unbroken skin and mucous tissue is the strong barriers and the strong defense against any microbes. Mechanical defense, microbiome. Now you know that microorganisms uh, they can uh, be they can protect their own biotope against pathogenic microbes. Chemical defense, it's some chemicals and enzymes in body fluids. Antimicrobial peptides, plasma protein mediators, and cytokines and inflammation eliciting mediators. And cellular defense are granulocytes and agranulocytes and lymphocytes. Yeah. Physical barrier. These barriers prevent the attachment and penetration of infection pathogens to the host body, and they are intact skin, mucous, beating of cilia, coughing and sneezing, flushing actions, urine, saliva, tears, vomiting, and diarrhea. Mechanical defense that physically remove it's a physical removal of pathogens from the body, preventing them from taking up residence, so preventing attachment by like tears. They wash their eyes and they eliminate everything foreign and and urine wash microbes out of the urinary tract as it pass through. As a result of the urinary system is normally sterile. Microbiome, resident bacteria of the skin, upper respiratory tract, gastrointestinal tract, and genital urinary tract compete with pathogens for cellular binding sites and nutrients, produce antibiotics-like substance, and by this way, microbiome, normal microbiota, um, keep its biotope uh, in keep its biotope in the normal condition without pathogenic microorganism, protect its own biotope against attachment of pathogenic microorganisms because all because normal microbiota take all binding sites and pathogenic bacteria they can't attach to the cell, and also they are firstly take all nutrients and they dominate in the biotope, and that's why pathogenic microbes are eliminated from the body. And also they can destroy pathogenic microorganisms by production some substance which can act like antibiotics, which destroy and inhibit pathogenic bacteria. Cell defense, uh, it can be realized by phagocytosis and also by killing with natural killer cells. Okay, phagocytosis, I hope you study in biology and histology, so it is not new information for you. And phagocytosis, it's a process by which particulate uh, such as bacteria are ingested in and digested by the phagocytic cells. So phagocytic cells, they can eat some bacteria, they can engulf some bacteria and destroy that bacteria inside the cell. So they can eat, they can digest some pathogenic microorganisms. Phagocytosis require energy which is generated through the glucose metabolism. It's one one form of the endocytosis. The other form is pinocytosis, which is the internalization of the fluid and solutions. And also, I want to say that phagocyte, it's a phagocyte are cells which clean our organism. They are cleaner. They destroy everything foreign. They destroy any garbage in our body. Phagocytic cell, they are neutrophils, are granulocytes that circulate in the blood and migrate quickly in response to local invasion by microorganisms. Monocytes is differentiated into macrophage when they migrate into tissue which resides in all body tissue. For example, Kupffer cell of the liver are macrophages. Histiocytes in connective tissue are macrophages too. And microglial cell in nervous system are macrophages too. So, two kinds of phagocytic cell, neutrophils from the blood and monocytes. They also present in blood, but in the case of infection, they migrate into the tissues and became the tissue's macrophage. So tissue macrophage, it's a monocyte which migrated to the tissue, into the tissue. So this is a process of phagocytosis. Firstly, macrophage need to be activated. Bacterial substance activates macrophage, and also some immune molecules like cytokines can activate macrophage. In the process of activation, macrophage keep energy, create um, uh, some some enzymes which is necessary for digestion, and so then it moves to the site of infection. It's a chemotaxis, and then try to engulf bacteria, try to engulf foreign particles. Uh, before engulfing, phagocytic cell need to recognize that it is foreign molecule, that it is not self molecule, that it's something wrong, something foreign, some garbage, some that our body not need this cell. Then, after recognizing as foreign as something foreign, macrophage engulf this bacteria. Then it stay in the phagosome. But phagosome does not have any enzymes to destroy bacteria. That's why phagosome need to fuse with lysosome. You know that lysosome, it's a organelles of digestion, and inside the lysosome there are a lot of enzymes which can destroy, which can digest, make a digestion. So after fusion, lysosome enzyme start the process of destroying of bacteria, and then release the molecules which are the product of the phagocytosis. Okay, look at this picture there. You can see that macrophages, they have some receptors like receptors for immune globulins, and also receptors for complement and other cell. Okay, neutrophils are granular segmented leukocytes, and they are predominant population of leukocytes, and 70% of their white blood cell are neutrophils. Function of neutrophils is phagocytosis. Functional purpose of neutrophil granule is to ensure fast and slow phases of bacteriolysis. That's why if in the blood in the blood analysis you see a lot of neutrophils, you should think about bacterial infection because phagocytosis in general it's a mechanism against bacterial infection. That's why the high level of neutrophils in blood analysis it's a sign of bacterial infection. Target of neutrophils are mostly bacteria, but also can be protozoan, fungi, and parasites, but not viruses. First to arrive at the site of infection, they are first to arrive. They first arrive to the site of infection. High number in inflammation, bacterial infection. Neutrophils have short circulation time, 7 or 2 hours, and being in the tissue 5 days. And neutrophils are small cell and have segmented nucleus. Okay, here we can see neutrophils, and here you can see segmented nucleus. Okay, monocytes and macrophage. Monocytes, they are 3 or 11% of the white blood cells. Circulation time is 1 2 days. They have large kidney-shaped nucleus, like that, kidney-shaped nucleus. And monocytes they present in the blood, but when they move to the tissue, they became a macrophage. And macrophage they absent in bloodstream, do not circulate, present in tissue 20-25 days up to several months years, and they are bigger than monocytes. Target of macrophage are bacteria, dead cell, and debris. Function: resident macrophage play an important role in maintaining homeostasis, cell growth, detoxification, exchange of lipids and iron, but their main role is the removal of dying cell. That's why I can say that macrophages, they are like a cleaner. They clean our organism against everything, every garbage like dying cell. Inflammatory macrophages are formed in the process of emergency migration of monocytes into the focus of inflammation. So in the case of inflammation, monocytes move to the site of infection and become the macrophage and take part in the inflammation. Okay, here you can see monocyte and its kidney-like nucleus. Movement of phagocytic cell. They can have amoeboid movement and chemotaxis. Amoeboid movement, the movement of the phagocytic cell in and out of the blood vessels and throughout the tissue in called diapedesis when they go to the site of infection. And chemotaxis is the movement of phagocytic cell toward our cell or organism by cytoplasmic streaming in response to chemical agent called chemotaxis. So some chemical agents can be like a sign that there are infection sites, and they call phagocytic cell to move here. Here we have an inflammation. Here we have an infection, and some molecules which called chemoattractants, they call the macrophage in the site of infection. And when phagocytic cell hears this, when phagocytic cells hear this voice, hear this call, they move to the site where infection, where chemoattractants stay. So chemotaxis, it's a movement to the site of infection according the call of the chemical agents. Okay, what can be like chemoattractant? So some part of bacteria like N4-methionine and like endotoxins, and also some like some immune molecules like complement, like arachnoid acid, fibrinogens, like histamine, like plate activating factor, like eosinophil chemotactic factor, and lymphokines. So look at this picture. Leukocytes in blood respond to chemical attractants released by pathogen and chemical signals from nearby injured tissue. Here we can see pathogen, and here we can see some wound with pathogen. So they broke the barriers, skin barriers, and they are present and they are goes to the tissue. And uh, and here we can see injured cell that secrete chemical signals into the blood that we have the infection, that we have the infections, that we have some pathogens in our organism. And blood immune cell, neutrophil monocytes, they begin to deapidases through these blood vessels into the site of infection. And also we can see the cell of resident macrophage, tissue macrophage, which first uh, which which first meet the pathogen, and this cell also produce some chemical signals to show other immune cells that we have an inflammation, we have the infection, you need to go here quickly. And then you can see immune cell catch this signal, catch this chemical signal, and immigrate to the site of injury and infection by deepidases from the blood vessel, and by chemotaxis they move to the site of infection. And neutrophils release cytokines chemicals from granulates into tissue. Within the damaged tissue, neutrophils release chemicals and that break apart pathogens. Monocytes differentiate into macrophage, neutrophils, and macrophage phagocytize pathogens and cellular debris. So they began to do its work. Opsonization. Opsonization is a process which helps phagocytic cell to do its work, because phagocytic cells need to recognize that it is something foreign, and sometimes bacteria can be encountered with the capsules, with some mucose substance. Uh, they can screen its antigens, and that's why sometimes phagocytic cell needs some help. And opsonization is the molecular mechanism whereby molecules, microbes, or apoptotic cells are chemically modified to have stronger interaction with the cell surface receptors or phagocytes and antibodies. This is the mechanism of identifying invading particles, antigens, by the use of specific components called opsonins. Opsonins are substances that bind to particles and make them more susceptible to phagocytosis, like that. Here, this is the phagocytic cell, and phagocytic cell try to recognize that it catch something wrong. And look here, uh, this is a pathogen, and this pathogen is attached by some opsonins like immune globulins, like complement proteins, like some other opsonins, and by this way, they can sign that this is wrong, this is foreign. They help phagocyte to recognize this cell as a foreign and make the process of recognizing quickly, fast, to be faster. And after recognition, after opsonization, then ingestion and vacuole formation. When contact with the particles is made, the phagocytic cell engulfs it, surrounded the particle with a part of its cell membrane. Once the phagocyte site engulfs the particles, the membrane enclosing uh, uh, the membrane enclosing the part the part particle pinches off and move into the cytoplasm of the cell, forming a phagocytic vacuole of phagosome. Lysosome, which are membrane-bound bags of enzyme fused with phagosomes and to form a phagolysosome. Then, okay, so here we can see the formation of phagosome ingestion and formation of phagosome. And inside the phagosome, pathogen enter to the inside the phagocytic cell. Then, uh, phagosome needs to fuse with lysosome because there are in lysosome everything necessary for digestion, like enzymes and toxic products uh, uh, of oxygen metabolism, which can destroy and kill bacteria. So after that process, it is necessary to fuse phagosome and lysosome and make a phagolysosome. And the process of intracellular destruction can begin. Inside the phagolysosome, the engulfed materials is destroyed by the hydrolytic enzymes in the lysosome. Lysosome contain granules of two types: primary granules, also called azurophilic granules because they stain dark blue with red stain. They contain many hydrolytic enzymes such as myeloperoxidase, lysozyme, and arginine-rich basic proteins or cationic proteins. These primary granules represent about 33% of all lysosomal granules. So they need to kill bacteria to destroy. And secondary granules represent about uh, 67% of all lysosomal granules, and they include alkaline phosphatase, lactoferrin, and lysozyme. So, uh, respiratory growth during phagocytosis. The metabolic events which during phagocytosis are result in the production of different number of toxic oxygen metabolites such as superoxide anion, hydrogenic peroxidase, sunlight oxygen, hydroxyl radicals, halide, chlorine ion, myeloperoxidase, and hypochlorite. And you know that toxic oxygen metabolites, they can destroy the membrane of the pathogen, membrane of bacteria. And that's why respiratory burst, it's a first step of the bacterial killing. And then enzymal digestion began. Outcomes of phagocytosis. Uh, it can be in a free ways. full phagocytosis. When when phagocyte destroy uh, destroy the uh, full phagocytosis, when phagocyte can engage and destroy the pathogen, and uncompleted phagocytosis, when they can be kept inside the phagocytic cell. They can survive like that, multiply inside the phagocytic cells, and they can move through the organism inside the phagocytic cell. Okay, in the case of uncompleted phagocytosis, disease will occur because phagocytic cells, they can't do its work, their work, and microorganisms stay inside the human body and cause the infection. Okay, and also natural killer cell, they part of innate immune response. Uh, Natural killer cells as mononuclear lymphocytes use non-specific mechanisms to recognize and destroy cells that are abnormal. The target cell uh, for natural killers are cancer cell and cell infected with intracellular parasites like viruses. Recognition of such cell involves a complex process of identifying inhibitory and activating molecules markers of the on the surface of the target cell. Molecular markers that make up the major histocompatibility complex are expressed by healthy cell as an indication of the cell. So all human body cell, except erythrocytes, they have main complex of histocompatibility first type, and this is a sign in this is receptor to show that this cell is self cell and it's normal cell. But in the case of viral infection, in the case of cancer transformation, cell will lose its MHC receptor, or MHC receptor will be abnormal, and the cell will be recognized as an abnormal cell and will be destroyed by natural killers. Natural killers are able to recognize normal MHC markers on the surface of healthy cell, and these MHC markers serve as an inhibitory signals preventing natural killer cell activation. However, cancer cell and virus infected cell actively diminish or eliminate expression of MHC markers on their surface. When these MHC markers are diminished or absent, the natural killers interpret this as an abnormality and are cell in distress. When natural killer cell recognize a decrease in inhibitory normal MHC molecules and an increase in inactivated molecules on the surface of a cell, the natural killer cell will be activated to eliminate the cell in distress. So, in on this picture, you can see normal cell which have normal MHC receptor, and you can see natural killers. Natural killers check MHC receptor and recognize the cell as a normal, as a cell, and doesn't kill it. But look at this picture. This is cell which infected with the viruses, and its MHC is damaged. MHC is not normal. And natural killers recognize this cell as infected, as not normal, and produce a specific substance which perforates, which destroys this infected cell membrane, and this cell is killed by the natural killers. And in addition to the intracellular destruction, phagocytic cells secrete various compounds that have a protective effect in body. Among these are factors of influence: cell differentiation, cytotoxic factor, hydrolytic enzymes, endogenous pyrogen, complement, alpha interferon, various plasma proteins, and others. Innate immunity, it is not only cell immunity, and not only cell immunity, and not only barriers, but also some chemicals which presence in our fluid. Numerous substances found in body secretion provide a natural defense against microorganisms that invade the body, like organic acids, lysozyme, acids in gut, bile acids, mucose, sialic acid. The antimicrobial peptides, a special class of non-specific cell-derived mediators with broad spectrum antimicrobial properties, like bacteriocins, histotines, and other. And also acute phase proteins, class of antimicrobial mediators produced in the liver and secreted in the blood in response to inflammatory molecules from the immune system, like C-reactive protein, serum amyloid A, ferritin, transferrin, fibrinogen, mannose-binding lectin. And complement system. The complement system is globulin type proteins which are mainly synthesized in the liver and make up about 5 of the globulin factor or fraction of the blood plasma. The complement system includes more than 30 components factor. Complement in normal condition, complement is not activated. To act is not fused, is not joined to each other, but they can be it can be activated by three-way: classical, lectin, and alternative pathway. And in the case of classical pathway, the activator factor is the complex of antigen antibody complex in the classical pathway. In the lectin pathway, the and alternative pathway, the activators will be the part of the bacterial cell, like lipopolysaccharide, like a teichoic acid, lipoteichoic acid. And after activation of the complement, the membrane attack, lytic attack complex will be created, and the cell will be destroyed. Bacterial cell or cell infected with the viruses. Okay, here you can see also the scheme of complement activation: alternative pathway, classical pathway, and lectin pathway. So classical pathway, it's always activation with antigen antibody complex. Alternative pathway and lectin pathway is activated by the microbes molecules, like lipopolysaccharide and other. Then activator occurs, all part of the complements joined together, and the last one is C8, C9, And in the result of complement actuation, membrane attack complex is created, and this complex attach the membrane of the pathogen, and the lysis of microbe is occurred. Components of the complement system can lyse many bacterial species. Complement fragments released in this reaction attract phagocytosis to the site of the reaction. So complement can work as a opsonins. Complement components opsonize the bacteria for phagocytosis, so they help phagocyte to recognize bacteria as a foreign. In addition to the response shown here, activation of the complement system increase blood flow and vascular permeability at the site of activation. Activated components can also induce the release of inflammatory mediators from the mast cell. Um, complement system function. So it's a killing, lysis of pathogen, opsonization, chemotaxis, inflammation, cell activation. Okay. Also, when we talk about chemokines of innate immune system, we need to talk about cytokines. Cytokines is a general term for a large group of secreted molecules involved in the signaling between cells during immune response. All cytokines are proteins and glycoproteins. Interferons, interleukins, chemokines, tumor necrosis factor, transforming growth factor are cytokines. When we talk about cytokines, we need to say that they are like messenger molecules. By cytokines, immune cell can talk to each other, can send the signal to each other, can interact to each other. So examples of the cytokines, it's interleukins, chemokines, interferons. For example, function of interleukins stimulate and modulate most function of immune system. Chemokines recruit white blood cells to the infected area. Interferons usually associated with antiviral response. Uh, Cell infected with the viruses, they produce interferons to show other neighbor cells that they need to make a defense against viruses. Inflammation eliciting mediators like histamines, leukotrienes, prostaglandins, and bradykinin. Okay, interferons, proteins produced usually by virally infected cell, and they protect other cell in the area. Also, there are other triggers for interferon release. Virus infection is the most common and natural one. Types of interferons, there are three types: alpha, beta, gamma. Alpha secreted by macrophage and other leukocytes. It is induced by viruses and synthetic polynucleotides. Beta interferon is secreted by fibroblasts, also by viruses or synthetic polynucleotides. And gamma interferon is also called immune interferon. It's secreted by T lymphocytes following stimulation with a specific antigen to which the lymphocyte has been sensitized. Protective effect of interferons. Interferons activate cell genes, inducing a neighboring cell to produce antiviral proteins that interfere with the translation of viral messenger RNA. So they stop the process of viral RNA translation, viral replication. Interferons block viral translation and replication. Okay, look at this picture. This is a viruses and infected cell. Infected cells start to produce interferons, and also T lymphocytes catch the antigen and understand that we have a viral infection and also produce interferons, and other cells which are not infected, they catch the signal from infected cell and T lymphocytes that we have a viral infection, and it became the virus resistant because it changed the genes, and it stopped the process of viral translation, and viruses can't multiply inside the cell. Um, okay, also signals neighboring uninfected cell to destroy RNA-induced protein synthesis. Signals neighbor infected cell to undergo apoptosis, activates immune cell. This is what interferon can do. Other protective action of interferons are to enhance T cell activity, activate macrophage, increase the cytotoxic action of natural killers. Okay, thank you for attention. If you will have any question, please ask me in direct.